All right, staggering punctuality. Thank you all for coming. I don't know if I have any housekeeping obligations, but if the flames start to lick at you, I would just run that way, and there's a door there. And this is George Davey Smith, one of the world's greatest living epidemiologists. I think there are lots of different things you can be good at in epi. There's being really sort of process-driven and technically insightful around the structure of the stats and the maths in the data. And then there's having a kind of cunning around spotting opportunities in the structure of the world around you that you can use and exploit to do reasonable tests, fair tests, sensible, reliable analyses of the associations between one thing and another. And infuriatingly, George, as well as leaving the room, George is unusual in being very good at both. And I think the fact that he latched on to Mendelian randomization early on is a real testament to that, a kind of hidden, buried part of the structure of the world that you can use as an opportunity to do reliable analyses, looking at variance beyond just the normal kind of handle cranking that people were doing before, but also with risks of its own handle cranking, which I'm sure he will pour scorn on as well. George Davey Smith.
So thanks very much for that kind introduction, and thanks very much for the introduction. Now, entirely my fault. I misread the time slot, and so I thought this was an hour-long time slot. So I'll go fast. But if you get hungry, I fully understand if people just slowly leave and get the sandwiches as soon as the sandwiches arrive, and you can bring them back down again, I guess. But it's entirely my fault, not the wonderful organisation that has occurred. So I'm going to run very quickly through Mendelian randomization, what it was, what it now is, and what it should become. I'll be going very fast during the what it should become. I'm going to talk more about the background to Mendelian randomization, which was the demonstrable failure of observational epidemiological studies when identifying things that prevented or might cause disease, and then these were tested in randomized trials where what was randomized was precisely the same as what was tested in the observational studies or looked at in the observational studies, and they got different answers. There must be something wrong. And this became the sort of notorious thing that it led to cartoons. There won't be anyone in this room, I doubt, who's not seen this cartoon before, but this is one Shah Ibrahim and I used in 2001 in a paper called Epidemiology, Is It Time to Call It a Day? You've got the newsreader presses a button, and then the three arrows spin round, and they come up with today's news, which is that coffee causes depression in twins, the sort of random news, random scares generated by epidemiology. And there's lots of combinations you can work through, all of which could have been headlines. It's very easy to see that only one of these combinations is true. And the one that's true is that red wine does cause sexual dysfunction in men aged 25 to 40. But it's a very long time ago that I can remember such things. So Mendelian randomization tried to use a trick of using germline genetic variants precisely because they could not suffer from the conventional confounding that you get in observational epidemiology with socioeconomic factors, behavioral factors, most importantly, perhaps reverse causation, where early stages of the disease process lead to influence the risk factor that you're measuring rather than vice versa. And it was easy to demonstrate that you cannot statistically solve this problem in many situations. It's not solvable. And so therefore, you took a genetic variant which could not be confounded and obviously couldn't be reverse causation. Developing disease can't stop what you got allocated at conception, can't change what you got allocated at conception. So if you had a genetic variable in the LDL receptor gene which influenced LDL cholesterol, then that wouldn't be confounded. There wouldn't be reverse causation, although that could happen for cholesterol levels itself that were measured. And if you looked at the joint association of the genetic variable and the outcome at the top, and then you divided that by the association of the genetic variable to the modified exposure, then you got your estimate of your log odds ratio or log relative risk, depending on your design, for the causal effect. That was the notion. Now, the fundamental assumption of this is that it doesn't matter whether your cholesterol is influenced by the fact that you're eating too many sausages or is influenced by the fact that you were assigned genetic variants at conception that led to elevated cholesterol, compared to a counterfactual where you'd got a different selection of variants from your parents and had lower cholesterol. It doesn't matter whether that difference is due to those modifiable factors that we want to understand or is influenced by genetic variation. The downstream effect on coronary heart disease is the same. And this assumption, which in causal inference is now known as the consistency assumption, and it's fundamental to causal inference. In Mendelian randomization has a different name of the gene-environment equivalence assumption. This is that the effect is equivalent whether it's the environment or genetics influencing your exposure. And I'm hopeless at the technology, you see. I'm desperately pressing. Oh, no, it's worked now. There are many examples in monogenic disorders, for example, where gene-environment equivalence is definite or is observed. So, for example, pellagra on the right is caused by long-term deficiency in niacin, vitamin B3. And there's a rare monogenic disorder called Hartnup disease, which looks like pellagra, and is caused by a genetic disorder which results in poor absorption of amino acids, including tryptophan, which is converted to niacin. And you get the same phenotype. There's a differential diagnosis. The same output is seen. And so this reflects what the phenocopy-genocopy dialectic, i.e. that genes can imitate the environment, like a genetic variant in the LDL receptor can mimic having hypercholesterolemia because of your diet. And the reverse of that is, of course, true. It just depends whether you think genes are first or environment's first. So in the Soviet Union, Schmalhausen called it genocopy because the genes were copying the real thing, which was the environment. In the US, Goldschmidt called it phenocopy because the environment was copying the real thing, which was genes. But it means the same thing. And Zucker, Candle, and Villett famously said, no doubt all environmental effects can be mimicked by one or several mutations. So I do think that for endogenous disease processes, this is true. I think being hit by a truck, I don't think you're going to get genetic variation which mimics some things, but probably most things that are related to endogenous disease, it's true. And so, and here, for LDL cholesterol, we sort of know it's true because one of the environmental factors, of course, that now explains probably a reasonable amount of the variance in cholesterol in populations is taking statins. And genetic variants in the HMG-CoA reductase gene that relate to higher LDL cholesterol relate to higher risk of coronary heart disease, and statins are HMG-CoA reductase inhibitors, which inhibit HMG-CoA reductase gene, and they lead to lower cholesterol. So obviously the gene and the environment are equivalent here, and the same is true for PCSK9 inhibitors and PCSK9 variation, etc. So, and this is data from drug trials and genetic studies of these same, I'm not going to go through them all, but these are the same situations where cholesterol-lowering drugs target very specific genes which influence cholesterol level. Now, it's really important, this graph is really important because, of course, they're not equivalent because you start taking a statin at 50, but your genetic variants exist from the time of conception. And we know that atherosclerosis is a cumulative process. It builds up during life, And so the genetic variants mean you're exposed to something from conception on, essentially. And so, of course, the genetic variation estimates a larger effect because it's talking about lifetime exposure rather than just taking a statin for 5 years in the randomized trials, for example. And this is useful data because it shows that you get about 40%, 35 to 40% of the possible benefit within 5 years, which is impressive. It's an impressive benefit, an impressive proportion of the benefit that you're getting. But it also shows the potential of primordial prevention, that reduced cholesterol from birth onwards would reduce coronary heart disease risk even more. So gene-environment equivalence requires justifying in all situations, and it relates to biological processes that are influenced by the genetic variation. And so let's consider body mass index or adiposity as the phenotype. And so higher calorie intake. Now, FTO, which was the first genetic variant that was reliably related to higher adiposity, higher body mass index, Tim Frayling, Nick Timpson et al., 2007, demonstrated that. It's a famous science paper. Nick Timpson then went on to show that in ALSPAC, the Avon longitudinal study of parents and children, the kids who carried the 2 copies of the larger, greater body mass index variant ate more, just consumed more calories. And similarly, physical activity level, you would think, genetic variants are influencing BMI through physical activity. That's the same as you'd want to do with the modifiable exposures. Now, in a sort of famous quip that sometimes people have taken too seriously, Miguel Hernan pointed out that one very good way of reducing your body mass index is to saw one of your legs off. If I sawed one of my legs off, my body mass index would fall reasonably, instantly. But it's unlikely to make me live longer. And of course, there's no genetic variants which match to losing a leg. That's not instrumentable. So those things aren't instrumentable. But then, God, I'm really crap at this. But then, and then I put a question mark, but I think there isn't any question mark here, which is smoking. One of the variants, which is genome-wide significant for body mass index, is a variant in a nicotine receptor gene, which relates to smoking more heavily if you smoke. If you start smoking and you carry this variant, you get much higher cocaine levels, even at the same level of cigarettes, because you're inhaling more, probably smoking further down, closer down the butt, etc. And, at the cost of £10 million, the first GWAS of lung cancer, this was the top hit. So the first genome-wide association study of lung cancer that got a genome-wide significance hit found this variant. So at the cost of 10 million quid, Mendelian randomization showed that smoking does indeed cause lung cancer. And for lung cancer, the genetic variant has gene-environment equivalence for lung cancer. But for body mass index, it doesn't. For body mass index, if you smoke more heavily, you have lower body mass index. People know this, by the way, which is one of the reasons why smoking is used as a way of maintaining weight. And also, Mendelian randomization shows that your BMI does actually relate to more smoking. Maybe, we don't know what the mechanism of that is, whether it is entirely through voluntary attempts to maintain weight, or whether there's some biological mechanism in terms of you metabolize the nicotine more and you want to get more of it into you to get the same hit. We don't know that, but we do know very reliably that BMI influences smoking behavior. It makes you smoke more, higher BMI. So here, of course, if you are trying to instrument BMI, even though this variant is a genome-wide significant hit for BMI, you would be pretty stupid to use that as a genetic variant for BMI when you're looking at the effects of BMI on other health outcomes because it relates to heaviness of smoking. In the case of lung cancer, it almost certainly is entirely reasonable. So a pin-up early example of Mendelian randomization was in relation to C-reactive protein and coronary heart disease. So C-reactive protein is an acute phase reactant. anything bad that happens to you basically increases your C-reactive protein, infections, smoking increases your C-reactive protein, etc. And it strongly predicts coronary heart disease. This was a collaborative venture now a long time ago, showing that adjusted for age, sex, and ethnicity, a standard deviation higher CRP was related to 50% higher risk of coronary heart disease. You adjust for all the confounding factors that you can measure, and that goes down, from half to a third higher risk, but it's massively statistically robust. And CRP was starting being called the new cholesterol. Pharmaceutical companies were trying to develop drugs to inhibit CRP because they thought this would reduce coronary heart disease risk. So what you see, this is CRP measured. So this is the phenotype, the protein circulating in the blood. And what you show is that it relates, as I said, it relates to everything bad. Being old's bad, honestly. Being older relates to higher CRP. Having higher BMI does, systolic blood pressure does, smoking does, lower socioeconomic position and deprivation does. Everything bad relates to it. But Mendelian randomization does indeed do what it says on the tin. I really can't change. Oh, there we go. Sorry. I'll stop whining. So here's the genetic variant, which is in the promoter region of the CRP gene. And that genetic variant relates to CRP level, to circulating CRP level, but it doesn't relate to any of those confounders. So this is doing what it says on the tin. The genetic variant is not associated with any of the conventional confounders. And you'd be possibly unsurprised to hear that when you do the Mendelian randomization analysis of C-reactive protein and coronary heart disease, it's totally null. And pharmaceutical industries that were developing CRP inhibitors stopped developing CRP inhibitors. I think possibly the most important thing to get from this slide, though, is that what you see at the top, that is what you see in conventional epidemiology when there is no effect. You see a reduction from 50% higher to a third higher risk. That is what you see with heavily confounded factors, with reverse causation in process, after you do all the statistical adjustment for a vast number of things. That's what happens. That's an important take-home message and why observational epidemiology led to the wheel-spinning cartoon, because that's what people were presenting. So you can do Mendelian randomization in two directions. I said that BMI relates to higher CRP. What's the direction of causality there? Well, using this FTO variant, Nick Timpson showed that indeed BMI increases your CRP. So that shows that BMI is a confounder, because remember, to be a confounder, a factor has to cause your exposure of interest and has to cause your outcome. And BMI obviously does indeed cause CRP and causes heart attacks. The other way around, CRP on BMI was completely flat. Probably another sort of pin-up early example was HDL cholesterol. It was known as good cholesterol. I did publish a paper with Andy Phillips in 1991 talking about the measurement error in confounders, saying that we thought the evidence wasn't strong, and doing a whole load of modeling on this. But pharmaceutical industry spent $1 million on trials of trying to raise HDL cholesterol, thinking it would reduce coronary heart disease, through a whole set of mechanisms. None of them did. Now, I think an important thing, and I think the thing I hope Mendelian randomization will go back to, is Mendelian randomization is not just about estimating instrumental variable effects. You can have qualitative evidence from Mendelian randomization that can be really useful. A lovely early example without an instrumental variable was in sheep dip. Some sheep dips contained a chemical which farmers thought were giving them symptoms, but they were generally sort of malaise, fatigue-style symptoms. Was this the case? Was the sheep dip itself doing it? And so Nicola Cherry did a lovely study where she looked at a genetic variant in peroxinase which would detoxify the sheep dip. And she just looked in farmers who had symptoms and those who didn't. And indeed, the ones who had symptoms were less likely to carry this genetic variant. Now, there's no IV. There's no genetic variant for using sheep dip. The problem is that if you GWAS, if you did a genome-wide association study of using sheep dip, I guarantee you would get genome-wide significant hits, and you could attempt to carry out Mendelian randomization. And sadly, that sort of study is what's filling the literature. And this was, I think we had to keep repeating this in the first paper, that we're not saying screen people for this gene and don't let them work as farmers if they've not got the gene. We're actually saying that this tells us at a population level about the cause and effect of a particular exposure on health outcomes. And there's, another example is there's a slow acetylation genotype which leads to slow detoxification of a carcinogen in smoke, that's in tobacco smoke. It does not relate to smoking behavior, but it relates to the detoxification. It is not an instrumental variable for smoking. It doesn't relate to smoking, so it can't be. But the fact that genetic variant shows an association with bladder cancer in the slow acetylators, but not in the rapid acetylators, is very powerful evidence that smoking is causing bladder cancer. So what types of Mendelian randomization study are there these days? Most Mendelian randomization studies now are carried out as two-sample Mendelian randomization, where you get the genetic data for your exposure from one GWAS and your genetic variants for your outcome in the other GWAS, and you divide one by the other, and that's it, and then you've got a paper. The GWASs are easily available. I mean, literally, that's it. And I call these Noodles for reasons I will come on to later. Then there's Mendelian randomization, which is one sample, which is where you get the genetic variation and the exposure, the genetic associations to the exposure and the outcome in the same sample. Now that is fine, except you suffer from something called winner's curse, which is that you overfit the data. If you're doing both things in the same sample, you overfit it. So CFANG developed a jackknife approach where you sort of basically you GWAS 90% of UK Biobank, because that's the only sample people do this in these days, UK Biobank, and then you apply that to 10% on the outcome data where they're completely independent. You do it 10 times and you meta-analyze them, and then you don't get the overfitting. So you can do one sample MR as long as you use a jackknife approach. Then there is individual data, which is where you use summary data for the exposure, and then you use individual data for the outcome, and that is UK Biobank, basically. People now use other GWASs to get your instruments, and you apply them to UK Biobank. And then there's not, the other corner doesn't exist. So where did the name come from? So this was a letter we wrote in 2001 in response to a critique of a paper we'd published on homocysteine and coronary heart disease. In those days, there was a lot of enthusiasm, particularly from some quarters, that folate was going to reduce coronary heart disease risk because it lowered homocysteine, and homocysteine was toxic. Homocysteine is like CRP, everything bad puts up your homocysteine. We argued using the 1991 stuff that I'd done with Andy Phillips, that measurement error in the confounders very likely explained the associations. The enthusiasts didn't like this and wrote in, and then we just wrote this letter where we did a two-sample MR calculation. I misspecified the standard errors. I didn't understand MR was an instrumental variable approach at this time, so I didn't use the proper methods. And I misspecified the standard errors a little bit, so you can write in and say, you can recalculate it, and the standard errors should be slightly wider. But from that we said, well, this throws some doubt on this. And then we said through Mendelian randomization, this provides the test with all the reasons, and the reference for that was a paper by the sadly late Richard Gray and Keith Wheatley, where they had used this term, Mendelian randomization, but in a very different context. In an absolutely ingenious way, they'd used this term in a model whereby you could get evidence of whether having a bone marrow transplant was good for kids with hematopoietic cancers. Now, parents weren't good, there wasn't any other treatment, so parents, understandably, didn't want to have generally want to have randomized controlled trials of something like bone marrow transplant, because it was about the only hope there was. So they had this brilliant idea that the most common source of a bone marrow transplant is your sibling, an HLA-matched sibling. So if the person had a genetically compatible sibling, they were much more likely to get a bone marrow transplant than if they didn't have a genetically compatible sibling. This was brilliant. The only one slight issue is they should have adjusted for the number of siblings, because the number of siblings influences the disease risk and might influence the outcome. But even so, this was a brilliant idea. And there had been other names, actually, which I only discovered years later. There had been other names for the same approach, which predated that. So Buckley et al. Referred to this method and referred to it as biological randomization. It turned out that Buckley had probably heard of it through Graham Wheatley, but got the paper out first. And it's been called meiotic randomization. It's been called genotypic randomization. Anyway, these are all the names. We chose Mendelian randomization because that was the one I'd seen, because I'd taught a course using it in the early 90s. So, and I think part of the issue with Mendelian randomization is some people don't like the name, but it's probably too late to go back to any of the alternative names. Now, the notion of it relates strictly to Mendelian and meiotic principles. So we introduced it as something that should be done within families, for example, between siblings, because if you do it between siblings, then it is literal randomization between siblings, and there's no population stratification, etc. There's no, and I'll go through the other biases you can get. And we said that between siblings was analogous to RCTs. That was the analogy, and we showed that. And then we then said there aren't any big between siblings. There was only one between sibling data set that we could look at, and I'll get to that example. They didn't exist, so we said, well, at a population level, the randomization is approximate. It's not true randomization, and then we still use the name. We probably should have should have stuck to AMR, approximate Mendelian randomization. And that would now mean AMR wouldn't mean that rather than antimicrobial resistance if we'd thought of that. But we didn't. Thomas and Conti said we'd demonstrated an instrumental variable approach, and we read that and thought, fuck, this is indeed an instrumental variable approach. But they told us, bizarrely, I had taught instrumental variables in the early 90s at the London School of Hygiene, but I was doing work on childhood diarrheal disease, etc. And economists were using distance from a water source as an instrumental variable for the amount of water you used. I was working in rural Nicaragua, and I knew that the distance from a water source related to 1,000 confounding factors. So the idea of that as an instrumental variables was bullshit. So I filed IV as shit. But I thought that Mendelian randomization was quite good. So I didn't even think about the fact you could use IV estimation techniques for it. But anyway. So this is the instrumental variable setup. You test that your instrument, here Z, or genetic variant in Mendelian randomization, has to associate with X, your exposure. Now that you can test. You can get a p-value for that. That is the only one of the three assumptions, the three principal assumptions you can test. The second one is exchangeability, or no confounding, between the instrument and the outcome. That's the second assumption. Now, I'd said, you did Mendelian randomization because of a lack of confounding, but the obvious confounder, which is possible, is ancestry. If you had a mixed population, half of the people from East Asia, half of the people not, then genetic variations that are prevalent in East Asia would relate to any disease which was more prevalent amongst East Asians if you didn't take that into account. That's called population stratification. Now you can take that into account with genome-wide association data by using the principal components, but it doesn't do it perfectly. What does it perfectly, or nearly perfectly, is looking at between siblings. So there can be confounding. And an important confounder, potential, which looks like a confounder, isn't actually a confounder, is dynastic effects through parental genotype. Because remember, you get your genotypes is like a 50% random sample of mum and dad's alleles at a locus. And if your parental genotype influences other things, like influences your exposure or influences your outcome, then you're in trouble. And this is the one example we had of between SIB, and I think it's still one of the most beautiful examples of Mendelian randomization, because this is for folate and neural tube defects. Now, we know from randomized controlled trials that folate supplementation in mothers reduces the risk of neural tube defects in the offspring. So here, you have a genetic variant which relates to folate activity, and it does now known to relate to folate level, in the MTHFR variant. And if your mother is homozygous, for that variant, you had a twofold risk of having a neural tube defect. If your father was, there was no serious elevation of risk. That's a beautiful negative control. This is combining Mendelian randomization and a negative control, because your father's genotype can't influence the intrauterine environment. That's your mother's genotype. But also, more importantly, if you just had the genotype in the kids, you would think that it's the kid's folate level rather than the mother's level. So you can see here how that confounding by what we call dynastic effects, which is the phenotypic effects of parental genotype. Again, we said don't screen, give people folate. So dynastic effects can be through the intrauterine environment and affecting the outcome, or it could be the parental environment. If your parents have genes which are related to higher educational attainment, there will be more books in the house. If your parents have genes related to drinking more alcohol, you'll grow up in a wetter environment and probably be more likely or less likely, one or the other, to drink alcohol, or same with smoking, etc. So you can get something which looks like, well, you don't get exchangeability. That is possible. So, and then the third assumption, but you can't test that. You have to use, like within SIBs, for example, to try and avoid it. The third assumption is that your genetic variant has no effect on the outcome, except that is mediated through the exposure. Now, one problem is people tried, just adjusting for the exposure. They thought, well, that's a test of it. There's a letter published saying we've got a fantastic test for the IV3 assumption in one of the JAMA journals. We wrote in about explaining why that wasn't the case, and also explaining that as there's been like 50 years of quite clever economists studying this, if you had got one, then you should get a Nobel Prize, or it's wrong. And so far, every one which has been advanced has been wrong, rather than Nobel Prize winning. So why does adjustment not work? Well, so here we can think of what now is known as collider bias. Adjustment doesn't work because when you adjust on something, like your MTHFR level or whatever, when you adjust for something, even if there's no confounding in the source population, you generate an association between your exposure of interest and other factors, which looks like confounding. It's actually through association, but it looks like confounding. And so an example of this that Jonathan Stern uses is to consider like at US colleges where people either get admitted on a sporting scholarship or they get admitted because of their SAT scores for their academic level. Now, let's imagine a world where academic level and sporting prowess are completely uncorrelated. There's no association. So, they're not going to influence what you're looking at. But then you decide to stratify, which, remember, stratification adjustment is just doing stratification without actually realizing you're doing stratification. If you stratify being admitted to college, and so you walk onto one of these campuses, you see a student, you throw a ball at the student, who tries to catch it, bounces off his head, falls over backwards. You can confidently go up to that person and say, "You are very clever." Because there's no possibility whatsoever that they have been admitted on a sporting scholarship. So those things become actually correlated minus 0.6 in that situation. But this is something which I think is probably underappreciated in epidemiology in general is that adjusting for factors induces associations even when they don't exist in the source population. So that's why you can't just adjust, as well as measurement error, known as collider bias. You've got to remember that all genetic variants that influence a phenotype have to do so through another phenotype. The gene doesn't have a direct influence on on exposures or outcomes that we're generally interested in. You know, you can get some very close molecular associations, but the sorts of things that MR is generally used for is not directly influenced by the gene. It's downstream of some level of processes. So it's through some unknown factor. So that's horizontal pleiotropy when you have another pathway between the genotype and the outcome that isn't through your exposure. That's horizontal pleiotropy. The problem is the term pleiotropy also applies to this situation where your gene influences your exposure, which then influences your outcome. That is called vertical pleiotropy, or Grunenberg actually called it spurious pleiotropy, but sadly that never took off. In 1938, called it that. Never took off. This, of course, actually is Mendelian randomization. Look at that's the figure that I was showing earlier. So if you imagine your genetic variant is FTO, X is body mass index, and Y is coronary heart disease, that is a Mendelian randomization experiment for looking at effect of body mass index on coronary heart disease. So Mendelian randomization is simply the use of vertical pleiotropy. Now, the problems with vertical pleiotropy arise when you don't know what the U is. So if U doesn't have an effect on Y, then that's okay. There always are U's. Remember, there's always a U. There's always gene expression, methylation, transcription factor activity, protein that you're not measuring, et cetera, et cetera, et cetera. There's some chain of these things in virtually every Mendelian randomization study you know. So you're making the bet that none of those things influence your outcome. Now, as I say, if they don't, then you're fine. But then imagine a situation, as I've already shown data on FTO and CRP, I'll use that as an example. Imagine Z is FTO, U is BMI, which you've not measured in your study, and X is C-reactive protein. Now, you could easily do this because the fourth most strongly associated genetic variant with CRP is FTO. FTO is massively genome-wide significant for CRP. So if you were just doing a naive Mendelian randomization using MR base or whatever, chunking through CRP, you're including FTO, using all the hits, you're including FTO as an instrument. And then you misspecify the primary phenotype and get the wrong answer. Now, you can do what's called Steiger filtering that Jip Hamani introduced, which is that for any downstream pathway, like FTO, BMI, CRP, except in extraordinarily unlikely circumstances, the variance in the thing which is closest to the genotype, i.e. the variance in BMI that is explained by FTO, is way greater than the variance in CRP explained by FTO. So you filter them, and to use a genetic variant as an instrument, it's got to be explain more of the variance in your exposure than in your outcome. And you can expand this to filter it against anything, because you could just sort of say, if this variant explains more in any factor than it does in the one that I'm wanting to use as an exposure, we shouldn't be including it. And that approach is being under investigation at the moment. But that's the situation with correlated pleiotropy, which is the biggest threat to Mendelian randomization. So when you've got IV estimates from MR, all of the IV assumptions need to be met. As I already mentioned, the exposure period is often not well defined, and I'll talk about this evening's lecture. It's often considered lifetime exposure, but people don't actually demonstrate that. You should go and actually show that when the genetic variant starts becoming associated with the exposure over the life course. Genetic variants associated with alcohol consumption don't become related to alcohol consumption, until 15 in Oxford or 13 in Warrington when people start drinking. But they obviously don't relate to alcohol consumption earlier in life. and may relate only to an exposure during a critical period. We have examples of when we think this is the case. Vitamin D does reduce the risk of multiple sclerosis quite markedly. A hypothesis is that it's the vitamin D level, though, at the time you get Epstein-Barr virus infection, which is necessary for multiple sclerosis, which relates to your response to it and whether that response becomes overactive and you end up developing multiple sclerosis. Giving people vitamin D after they've got EBV is no use. So Mendelian randomization relates to this whole window up until when the people are measured of exposure. But the exposure might only be acting at a very critical period. And if you've got categorical exposures, it may only be interpretable in terms of liability, not to the exposure itself. So imagine people often do MR saying prostate cancer against outcomes. But in their sample, virtually hardly anyone's got prostate cancer. So what you're actually testing is the genetic liability to prostate cancer, unless you have dated time when people develop the disease, and you can actually build that into your model. You could, in principle, then work out, do MR for getting prostate cancer. But so far, no studies have been done with that. And prostate cancer gives you a lovely negative control, of course, because you look in women and you look at how the prostate score relates to outcomes in women. I'm not going to talk about these. You'd be delighted to know there's a huge number of sensitivity analyses. There's a huge industry producing sensitivity analyses. Eleanor Sanderson reviewed them. I think one of the first ones introduced, I think, is in a way the most powerful, which is when you have zero relevance points. And I'm going to illustrate it with an example from on alcohol consumption. As everyone knows, whenever papers appear saying that alcohol reduces the risk of any disease, these are headlines. Everyone's enthusiastic about such papers. This is a recent one. 12 cardiovascular diseases, all of them were related and protected by alcohol. The Guardian loved it. Moderate drinking can lower risk of heart attack, says study. Going down market to the Independent, drinking a pint of beer a day linked to reduced risk of heart attack. The Daily Mail, further down market, cheers, drinkers have one glass of wine, less risk of heart failure now than teetotalers. The Daily Star, drinking alcohol slashes risk of heart problems. If you drink this much per week, that's the amount you could drink per week being held up. Sort of very sober US, The Time, alcohol is good for your heart most of the time. And my favorite one was the Irish Times, moderate drinking may cut risk of heart disease. That's a very good headline. Then below, moderate drinking may be good for your heart, and even heavy drinking may lower your risk of heart attack, a new study indicates. Now, this can be tested in East Asian populations quite well, because in East Asian populations there's a genetic variant which has a huge influence on alcohol consumption, which is in the acetaldehyde dehydrogenase gene. So to give a cartoon view of a metabolism of alcohol, ethanol is metabolized by ADH to acetaldehyde. Acetaldehyde gives you all the adverse consequences of drinking alcohol, so you get headache, you get flushing, you get palpitations, you get guilt. Actually, you don't get the guilt, actually, but you get all the other adverse consequences. And sensibly, people who have a knockout, a literal knockout, you don't have of your ALDH2 gene. If you're homozygous for that, drinking alcohol is extremely unpleasant. And here we have data. This is ancient, but I love this because this is the first one we did like this. The 2-2, that is the people who have 2 copies of the knockout. And the grey lines are men, and you see that even the men aren't drinking when they've got two knockouts. But in these populations at the time these studies were done, women simply didn't drink. They weren't drinking whatever their genotype. But for the men, there was a sort of stepwise change in alcohol consumption by the number of variants you have. So then if you're trying to say, does alcohol increase blood pressure, you have the perfect negative control. Because if there's any, or zero relevance point population, because if there's any pleiotropic effect of the genetic variant on blood pressure, for example, you'd see it in women. If it's not through the alcohol, the effect would be seen in women. But what you get is a perfect null in women. And in men, the bad news is that alcohol has a very substantial effect on increasing your blood pressure. So you've got to take an ACE inhibitor, if you're going to be drinking. Blood pressure needs to be watched. You can use it for estimation. I won't go into this. And this is a study Richard was involved in setting up many years ago, which used that zero relevance point, but actually applied it both to chromosomal sex and to region, because there's 10 regions in China with very different average levels of alcohol consumption, including Muslim areas with very low alcohol consumption. And you could combine those two to actually get some attempt to look at the actual shape of the relationship. And as you can see, the top is observational with stroke, and the bottom is Mendelian randomization with stroke. Sadly, there is no benefit at any alcohol level. This is a photo I took 30 odd years ago when I lived in Glasgow, and I used to drink Old Mortality, lovely beer, 80 shilling. And I can't sue them because I can't go back now and say you never, I've had the stroke because of you, because they were telling me all along what was gonna happen. So I talked about the sensitivity analyses. Now, as I say, there's an industry in these, and this is the template for an MR methods paper. The introduction says current methods have limitations. In the methods, you say, we simulate data and apply our new method and a handful of previous methods, selected previous methods, to these results. Our methods performed best. And the conclusion is we are the champions. Every single MR methods paper is like that. What we need is real-world examples like HDL cholesterol, where we know from trials the answers are wrong. And methods should be not just tried against simulated data, they should be tried against real-world data. And we should have banks of simulated data that everyone has to apply their method to the same, a bank of simulated data. Right, okay, MR, what it is. Okay. What are you instrumenting? God, I've been going on for fucking ages. Shit. People can go and get sandwiches, but so you've got to be careful of what you're instrumenting. So an example of genetically informed drug development is sclerostin inhibition, which increases your bone mineral density. This was entirely discovered through human genetics because there's a human genetics knockouts in the sclerostin gene, which lead to very high bone mineral density. And well, what we show is you do a GWAS, you get a couple of variants that are plausible variants, but none of them in the sclerostin gene itself, but there's some biology to explain, and the MR does indeed show that, higher sclerostin leads to increased fractures and to lower bone mineral density. But then you do a GWAS, and this is then getting onto the whole problem of the upstream factors of confounding, and now we get lots of hits, including one variant in SOST, which is the sclerostin gene, which is a very good instrument because it's very plausible that it relates to just local production of this in the osteocyte. What matters is the production of sclerostin in your osteocyte. You can't get human osteocytes to grow. So all the data come from circulating sclerostin in the circulation, which kidney failure increases that, etc. Lots of things relate to sclerostin in the circulation. And in particular, one large group of variants that influence sclerostin are variants that lead to higher bone mineral density. Higher bone mineral density leads through homeostatic mechanisms. Biology is all about homeostasis. Leads to increases in sclerostin and sort of attempts to downregulate it. You get loops, and so you get the very reverse of what you want to get. If you actually use these variants as instruments for sclerostin, you would get the exact opposite answer. So that's the problem of including all the variants. What you've got to think about is because you have homeostasis. Just remember homeostasis. And what you see is that the SOST variant, the single SOST variant, produces for the MR estimate produces a much larger effect, even though it's detected from circulating protein, the protein in the blood. You get a much larger effect than the other variants, which do actually relate to sclerostin, but not through homeostatic mechanisms which reverse the effect. So you exclude the ones that fail Steiger filtering. And that's probably because that variant will actually relate to the local sclerostin level that you can never measure in the osteocytes. That variant will relate to actually what you want to know. So just taking all the variants isn't a good thing. So what I'm going to do is I'm going to skip this section because I've just been going on for fucking ages. The bottom line is, if anyone's interested in this, there's some fantastically interesting, in my view, work on early life prepubertal body mass index, which higher levels protect you from fractures. And it's not your adulthood BMI that matters, it's your size in childhood. And for breast cancer risk, larger prepubertal body mass index is highly protective of breast cancer. And Mendelian randomization shows that. And it shows that there's two pathways, one through high childhood body mass index and one through age at menarche, which repulse each other. Because high childhood body mass index brings forward age at menarche, which increases the risk of breast cancer. There's two mechanisms. Those two mechanisms are independent of each other, and if we understood that, we would move some way forward in understanding the etiology of breast cancer. Okay, so the sad side of MR now, chronicle of a death foretold. When two-sample MR was introduced, Fernando Hartwig demonstrated by looking at just one paper, demonstrated errors that could be made in two-sample MR through not dealing appropriately with the genetic data. This led to the first retraction of an MR Mendelian randomization paper there was. They got the answers completely flipped. But we were saying that, this is too easy to do, but it's our fault because we put out tools that allowed you to do it. And in fact, Jim Hamani did this MR of everything versus everything, where we said we've done every single two-sample MR analysis you can do. You shouldn't publish two-sample MR analyses on their own because they've all been done. They're all available, and they are now all available through a thing called epigraph.DB. But sadly, a lot of two-sample papers get published, and this is the week-on-week publication. There's now 125 papers with Mendelian randomization in their title that are reporting MR results. This is a true exponential, and you can very easily work out that in 23.5 months, every single paper in the world will be a Mendelian randomization paper. So that's what's going to happen. These are the ones I call Noodles because, your GWAS... It's very sad, this paper, in a way, because this paper is well done. It's just that the gene-environment equivalence assumption is entirely erroneous. You can't GWAS noodles. People don't have a specific taste for noodles, which mean they eat more noodles. You've got so many... You're just looking at the upstream factors. And people actually GWAS whether you're on whether you're on atorvastatin or simvastatin. They GWAS that, and then they think, you get hits, and they then think, probably related to social position, you've got a crap GP. You should be GWASing the GPs to say whether they're giving atorvastatin or simvastatin. That would be more meaningful, not the people. Or you GWAS the postcode. For fuck's sake. You GWAS the postcode, and then you've got the air pollution data for the postcode level, and then you say it's an instrument for air pollution. It's ridiculous. This is a website in China, where you buy the papers essentially. It says, we produce all these papers. We give tutorials. Here's an advert. This is an impact factor 15.3. So it's very easy. I could easily identify this paper. I haven't put the paper up, which has been clearly not written by the authors of the paper. They're advertising by saying, look, we did this paper. We helped with everything about this paper. And the cost is 3,398 yen, and you do get discount. You can get a package of 5, and it's a lot cheaper than single ones. The student said, "I am not proficient in getting started just now. In the future, it will be done in minutes." And truly, it will indeed be done in minutes. And what happens with these factories is you get suddenly a new exposure comes out, and then within days you get all the gut microbial and the risk of erectile dysfunction. Anyone? There's like when I did this, there were 16 papers on, literally 16 papers of the microbiome onto erectile dysfunction, and they're not all published in ridiculous journals. So no Nulls. The tragedy of MR is that the important MR findings used to be CRP doesn't cause coronary heart disease. HDL doesn't protect against coronary heart disease. Many of the most important things were Nulls because not everything is a cause. You don't get any Nulls anymore. Now, not in the two-sample situation. And then the Numb Skulls are just complicated methods I shall just skip this, but are complicated methods which claim to be able to look at the association across the distribution. They are basically reflecting interactions between factors that relate to risk and the effect of your variant on your exposure level. That's why they fuck up, but they all totally fuck up. You might, it's not worth doing the nonlinear MR because you just get, as you can see there, those U-shapes, you get, as causal estimates, you get precisely the same shape as you see with the observational data. And this, here you get an overall effect 0.99, and every strata is substantially below 0.99. So you've got 4 stratas. Now everyone has this vague memory, I didn't hear about something called Simpson's paradox. If your estimates are causal, and the assumptions of MR apply, and the data are distributed as we know the data are distributed, this is impossible. So you just simply recapitulate observational findings. So it's really sad because vitamins were the crap that was produced. This is a reanalysis using a new method. This shows that if you do the MR and you look at chromosomal sex, vitamin D not only makes you, low vitamin D not only makes you die more, it actually makes you more male. And it makes you older. And the paper's retracted. The authors said, the authors say our results are logical impossibility. But remarkably, those authors and other authors carry on publishing papers using nonlinear methods simply because they are not absolutely impossible. They stretch credulity, but they are not, you can't demonstrate they're impossible by summarizing the strata. I find this unbelievable. This is a press release for one of these ridiculous papers. The sunshine vitamin that delivers on cardio health. And then this really makes me cry, honestly, nearly. It is not ethical to recruit people with vitamin D deficiency into randomized controlled trials and to leave them without treatment for long periods. Our genetic data, our Mendelian randomization data say you can't do trials. That is the exact antithesis of why we introduced this method. And it's deeply hurtful to me, personally. And this is bullshit about low BMI. This is, this might as well be the observational because it just looks exactly the same. And then you stratify by smoking. And I've just told you that smoking influences your BMI influences your smoking behavior, so it's a very clear total collider. Amongst never smokers, you don't see it, but amongst ever smokers. This is fantastic news for me. I am an ever smoker. I was quite enthusiastic ever smoker. My BMI is actually below the minimum BMI. I'm harming my health. I am harming my health by not putting my BMI up. And that's their conclusion. This is a BMJ paper that they have not retracted. The BMJ has not retracted this paper. Underweight participants with harm of having low BMI, harm in ever smokers, bit below my body mass index. So fantastic, knickknacks, here I come. 564 kilocalories per 100 grams, bring it on. I want to live longer. But remember, I've mentioned about smoking, don't smoke. You don't want to reduce your BMI by smoking, I mentioned that. But don't stratify on a collider is the message. And BMI, using the same methods we showed, BMI makes you more male and makes you more older. And of course, HDL cholesterol, the pin-up of useful MR, has been rescued by nonlinear MR. Nonlinear MR shows the same bullshit as you saw for vitamin D. Do not use these nonlinear MR approaches. And the reason for the 3 things I said, I think, is this increasing GWAS size underlies these, because you're just picking up more and more. As the GWASs get bigger and bigger and bigger, you're going to, more and more of the findings you get are going to be upstream factors, like BMI influencing CRP. But they're going to be upstream factors you don't know about. They're going to be molecular phenotypes that you can't even hope to measure. And so you just, and when you do that, you just recapitulate the observational associations. That's the beautiful thing with the cis variant and CRP. Does what it says on the tin. And Dan Rousseff, I don't know if Dan's here, probably not. He's a PhD student in Oxford. He's got better things to do. Dan Rousseff generated the current CRP score. It just relates to all the confounders. So the currently done MR relates to the confounders. So consider CRP as the phenotype, cis-CRP variants, yes. BMI, FTO and BMI. Smoking-related SNPs relate to, obviously not, they don't satisfy gene-environment equivalence. IL-6 absolutely certainly doesn't satisfy gene-environment equivalence. Anonymous trans variants are just, not cis variants, are unlikely to. So this is the problem with the multiverse, MR with the vast GWAS. I'm going to stop. I'm going to stop there. Oh no, I'm going to stop. Can I show these three more? They're very quick. So I am the Gerald Ratner of Mendelian Randomisation. So I think Ben's about old enough, Richard certainly is, to remember that Ratner famously got pissed at a meeting. He was a jeweler, doing quite well he got pissed at some dinner, and he started going on about how crap his jewellery was. People say, how can you sell this for such a low price? I say, because it's total crap. We even sell a pair of gold earrings for under £1. Some people say that's cheaper than a prawn sandwich. I have to say the sandwich will probably last longer than the earrings. So I am now being the Gerald Ratner of Mendelian Randomisation and saying what's currently produced in the warehouse is bullshit. I really actually rather like Gerald Ratner, who reinvented himself, and he said this about Starmer. I offered Starmer free jewellery, but he didn't want it. Even Starmer had some limits to his avarice. Thank you very much. Sorry, I went on so long. Sorry.
I'm guessing no questions.
No questions.
All right.
You can ask me questions up there.
If you want to ask questions, come back for the lecture at 5 and do it after that. Or come and pester us after food.
One question, Ben.
God.
You talk so bloody fast. What would be the single best reference, one best reference for somebody who wanted to read about the things you've just said?
So I dictated an email to you, which I have not sent yet, where I give the two references. There's a Cold Spring Harbor Perspectives in Medicine paper by Rebecca Richmond and me, and that's about 60 pages long and is really basic. It's basic, and that's but then the follow-up paper is by Eleanor Sanderson, is in Nature Reviews Methods Protocols, and that does all the technical stuff. So you read there's some overlap, but hearing things twice can be quite useful, and so those two references. So Rebecca Richmond and Eleanor Sanderson. I'm embarrassed to flick through all my slides because I put them at the end, but I'm so embarrassed to flick them through when to show how much more I had to say. It's shocking, isn't it? And thanks, I must say thanks to all these people. Although the fact that Dan's not come, I'm going to remove his name from the... that's Eleanor Sanderson's paper. And Nature Reviews Methods Primers. And did I put the book up? Yeah, on this one, if you, this is the Cold Spring Harbor. They're papers on open access, Rebecca's paper. But if you've got anyone in your family, friends that you really dislike, but they've got a birthday or Christmas is coming when Christmas comes, then you buy them this book. I guarantee this book will be the perfect present for the people you don't like. But it's in there, but it's available online. The paper, the single chapter's available online. Richmond and Davey Smith in there. See, I'd anticipate.
Give those two references to Ben, so you put them on the same website as the advertisers.
Oh yeah.
Okay. Yeah.
Right. I'm going to give George Davey Smith a sandwich. It's very nice to see you all. See you all at 5, hopefully, and thank you for coming. And thanks again to our philanthropist, Peter Bennett, sponsor of the Bennett Institute for Applied Data Science. And thanks to George Davey Smith. Bravo.